Hello, I want to talk to you today about DNA condensation because I believe that DNA condensation plays a major role in understanding epigenetics. Actually, DNA condensation is at the base understanding of epigenetics because how genes are expressed or not expressed, or, in other words, how genes are turned on and turned off will really be based on how accessible these genes are to proteins that will transcribe it and move it from gene to protein. That's specifically about DNA condensation. So if the DNA is really condensed, then sometimes genes may not be able to be expressed. Or if it's really open, then all of a sudden, genes can be expressed. And so we're going to talk about this whole thought of expression. And so just to kind of kick off, gene expression is the ability of a gene to be transcribed and then translated into a usable protein. It's a gene being transcribed, so going from DNA to RNA, then translated, going from RNA to protein, and that protein being functional. Several different things that can factor into what makes a gene able to be expressed or not expressed. But one of the major things in terms of epigenetics is DNA condensation. So let's hop into it. All right. So by the end of our talk today, you're going to understand or be able to describe the structure of a nucleosome. And we'll knock that out in the first few slides. You'll be able to get that so quick. After that, it's to associate the levels of DNA condensation and their impact on gene expression. And so when you see, you'll know exactly, okay, well, if it's tightly packed, what does that mean? If it's loosely packed, what does that mean? And how does that have an impact on gene expression? And I kind of gave you a clue earlier, but no worries. You'll see it again in just a second. And then finally, to relate the differences in the structure of euchromatin and heterochromatin and when and where they are found during the cell cycle. And so let's start off. And just to give you everything from the very beginning, I'm going to walk you through DNA condensation. And then we'll start to break down its role in epigenetics. If I were to go into one cell, pull the DNA, all the DNA inside that one cell, and stretch it out, it'll be as tall as a 6-foot man. If that's the case, how in the world does all that DNA find its way inside of the nucleus of a cell? Well, the way that happens is through DNA condensation. And so what you'll see here is that process of DNA condensation. Start off with the DNA. That DNA is then wrapped around these proteins. And this protein is very important when we talk about epigenetics because this protein or these group of proteins are called histones. So if you're writing things down, this is the first time to take some notes, these are histones, right? And this is an octamer. If you look, there's 8 parts to this. It's an octamer. It's to each of these 4 histone proteins. H2A, there's 2 of H2A, there's 2 of H2B, there's 2 H3, and then there's 2 H4. And that octamer is here, and DNA wraps around it. If you notice, DNA wraps twice around each one of these histone octamers. And if it wraps twice around, that means that there's going to have to be a lot of them in order for all this DNA to be really, really condensed, then find its way inside of the nucleus of a cell. This is the levels of DNA condensation. It starts off first with beads on a string, and it's DNA wrapped around DNA wrapped around these histones, DNA wrapped around the histone, but then there's some space and some looseness in between. By the way, this DNA wrapped around a histone, whenever you see that, that is the structure of a nucleosome. And I said we would learn that very early. Well, that's another write down part. You can write that down as gold. It'll pop up on the exam. It's DNA wrapped around a histone. That's the nucleosome. All right, so you have DNA wrapped around a histone here, and then this first formation is beads on a string. Well, as the nucleosomes get closer together or more tightly packed, then you're going to see this chromatin fiber. It's a 30 nanometer chromatin fiber. And this is now the stronger packing of this DNA. Now, here's the thing. Let's talk about one thing that you'll need for later. why does DNA wrap around these histones? What makes this interaction so strong? Well, the histones that you're seeing here are positively charged. And so histones are positively charged while DNA is negatively charged. And that attraction between that negative charge and that positive charge brings them together and allows them to pack very tightly with one another. And so, first beads on a string, then 30 nanometer fiber, and then there's more packing of that chromatin. The more packing of the chromatin leads to condensed chromatin, and then 700 nanometer chromatin. And then as it packs even more, then you get the chromosome or full-fledged chromosome. And this is what we're used to seeing whenever we're looking at like things like karyotyping or the cell cycle, and we see a cell, basically all the chromosomes lining up. This is kind of what you're used to seeing and what you've probably seen in the past. Now, this interaction between DNA and histones, or DNA and protein here, plays a major role in epigenetics. And so let's define epigenetics. And we haven't done that yet, but let's do it. So if we break down epi and genetics, the epi means above. So when you talk about epigenetics, it's above genetics or above the genome. And that's key because what it means is that I'm gonna just kind of help you to remember that, right? Epigenetics is above genetics, above the genome. And so it means that there are factors that are outside of the nucleotides that can play a major role in how genes are expressed, how genes are either turned on or turned off. And through epigenetics, you'll see how the environment plays a role in how genes are expressed or not expressed. And so epigenetics opens the doors for understanding of how stress could possibly play a role in gene expression. Stress causing some genes to be turned off, some genes to be turned on, even into adulthood, even into early childhood. How could stress do that? We'll explore that as we go through more of these talks about epigenetics. Diet plays a role in how genes are expressed or not expressed, and it gives some control back to you, because some people, kind of look at DNA as like the genetic lottery, if I kind of can say that, right? Whereas your genes are what they are, and you have no ability, based on either you have mutations or you don't have mutations, something like that, but you don't have a lot of ability on how genes are being expressed or not expressed. Well, through epigenetics, we understand that right now, stress can cause genes to change in expression. Diet can cause genes to change in expression. Changes in the environment can cause genes to change how they're being expressed. Exposure to toxins and trauma, and we'll explore that too, right? How can trauma play a role in how genes are being expressed? So genes are being turned off and turned on. What does that mean? Well, traits are changing, right? Traits are changing. Proteins that are being produced are changing based on outside factors having a role and playing a role in how your genes are being expressed. So that's what epigenetics is. It's very interesting. Understanding epigenetics is a very, very interesting thing, right? And we're going to go through the bases. And I feel like to understand all the cool stuff about epigenetics, understanding the basis of DNA condensation matters. We'll start off just kind of at this concept, right? So, do all somatic cells have the same DNA? Answer is yes. Yes. Every single cell, every single somatic cell in your body has the exact same DNA. All the genes that are in cells that are in your eyes are in, your stomach. And we'll talk about that in a second, right? So all the genes that are, available to your skin cells are available to your hair cells or brain cells or any other somatic cells. Somatic cells are just the cells outside of your sex cells. And so all these other somatic cells have the same DNA. Yes, they do. Now, the question is, well, why is my eye different from, my gut? Or why is, the heart different from the liver? right? So if they all have the same exact DNA, then why is everything so different? And the answer to that question is really in what's being expressed, which proteins are being made in each one of those spaces. And so the proteins that are being made in your eyes are different than the proteins that are being made in your stomach. So, and then, yeah, how are your heart cells and skin cells and neurons all different? It's based on which genes are turned on and producing certain proteins, and which genes are turned off and not producing certain proteins. All right, so their gene expression differences. Gene expression is different in the lung versus the pancreas, heart versus the red blood cells, skin versus your neurons. So, give an example. Red blood cells need to make protein hemoglobin so it can carry oxygen. Is hemoglobin a protein that's made everywhere in every one of your cells? No, right? But it's needed in your red blood cells. And so those genes to make the hemoglobin are turned on in red blood cells and may be turned off in other places. Your neurons need to make proteins called neurotransmitters to send information to other cells. Those neurotransmitters may not be turned on in every other place. However, they are turned, the genes to make the neurotransmitters are turned on in your neurons because it's necessary there. So this whole concept of why, if all of these cells have the same exact DNA, why is everything so different? Well, it's also different because of how those genes are expressed. So gene expression is the process of turning on a gene so that a protein can be produced. Gene expression must be regulated though, because not all genes can be turned on and off at the same time, right? Necessary proteins must be synthesized at appropriate times. And so it just means that at different times of the life cycle of a cell, different proteins need to be made. So there must be some regulation in when to turn the gene on and when to turn the gene off, right? All of that matters. And this is just, something funny I saw in a video where you have a eye cell, right? It says, hey, are we supposed to be making anything? This is the eye cell said, yeah, it looks like we're supposed to be making hydrochloric acid. And in the eye cell department, they're sitting there making this hydrochloric acid, making the hydrochloric acid. And then it says, wait, does that say stomach? Wait, is this not the stomach? And they're like, right. Because basically your eye cell has accessed the genes to make hydrochloric acid, and they start actually making that protein. And all of a sudden it's a very unpleasant feeling happening in the eye because the wrong gene was turned on in the wrong place. So the expression is off, right? And so you don't want that expression to be off. That expression being off could be a problem. And if you want to see this like really play out, you can look at the Amoeba sisters. They did a really good job of this. So this relationship between gene expression and DNA condensation really matters. And so I'll give you just a brief stuff, and we'll actually dive into this a little bit more in one of our next lectures. But to first start off, if a DNA is fully condensed, then the protein needed to transcribe the gene can't access the DNA. And I'm going to show you a very clear example of that, right? And if you remember which proteins are involved in transcription, and I don't know if you remember, but hopefully you do, RNA polymerase 2 is involved in transcription. And because RNA polymerase needs to have access to the promoter in order to transcribe a gene, it needs to have access to the gene in order to transcribe it, it can't get in there if the DNA is too tightly packed. Remember, DNA and histones tightly packed because of that negative and positive charge. So if the DNA is fully condensed, then the protein needed to transcribe it can't access the DNA. Chromatin. Chromatin is the association of proteins with DNA. And we showed you that. We showed the actual structure of the nucleosome. Chromatin is that association of proteins with DNA. And the name of those proteins are histones, because we talked about that. All right, chromosomes are DNA molecules and their associated proteins that are folded into a compact structure. So once you have DNA wrapped around those histones, you see, you start packing the nucleosomes, packing of nucleosomes. They get further and further. However, after the nucleosomes start packing up, you start to see that rope-like structure, and the folding of that structure over and over again creates the chromosome. Now, what are the types of chromatin? Well, there's euchromatin, more expanded DNA, and less condensed. And so euchromatin, and you can see the E in the euchromatin goes with the E in expanded. So euchromatin is more expanded DNA and less condensed. Heterochromatin goes with the H in highly condensed DNA and more DNA packaging. And so when you have loose chromatin, euchromatin, highly condensed chromatin, heterochromatin. So depending on the gene, an individual DNA strand can have areas of heterochromatin and areas of euchromatin. So you'll see, based on where it is, right? DNA strands can have some areas where you have more expanded DNA and some areas where you have highly condensed DNA. Genes in euchromatin, where things are more expansive or expanded or less condensed, can be expressed. It's highly condensed, no expression. If it's loose, high expression. As we get into this more, you'll start to see where that plays a major role in gene expression and which chromatin modifiers. So there are proteins that can cause tightening or cause euchromatin. There are proteins that can cause heterochromatin. And because there are proteins that can cause these two distinct states of chromatin or distinct types of chromatin, those chromatin modifiers matter. And we're going to learn about those as well. All right. So give an example. If there is a red blood cell, does it need to express neurotransmitters? No, we talked about that earlier. Well, neurotransmitters are not needed in red blood cells, but they are needed in neurons, right? So if there's a neuron, does it need to express hemoglobin? No, does not, right? So in those particular cases, those genes will be turned off. They can either be, and they'll most likely be turned off through what we talked about, which is this heterochromatin, euchromatin. All right. If a cell never needs to express a protein, it can be kept in the heterochromatin state. And that way, those genes will never get turned on. So if you take nothing else away, you should take away loose chromatin, high gene expression. Heterochromatin or highly dense chromatin or highly condensed chromatin, no gene expression. Loose, yes. Tight, no. If you walk away with that from this talk, that's great, right? So what about the gene for helicase? Does it always need to be expressed? This is a great question now, right? Because now we're starting to understand exactly when things need to be expressed versus when things need not be expressed. So the gene for something like helicase, when would that need to be expressed? I'll give you a second to think about that. When would we ever need that gene, this helicase gene? When do we need that, right? Well, helicase is involved in a part of the cell cycle, in the S phase of that cell cycle. And so because helicase is involved in the S phase of cell cycle, it's when we, use helicase to unzip DNA. What are two major times when genes are packed into heterochromatin? One is to silence gene expression. And that example of hemoglobin neurons is one of those examples that we keep using just because it makes a lot of sense. And it'll help you recognize and realize that. Two is to pack DNA tightly up for mitosis. So, and I told you, you probably recognize chromosomes from a particular time in the cell cycle, and that time is metaphase, right? Because you can see it at the metaphase plate, and you see that really, really clear in mitosis. And so when we want to silence genes, heterochromatin. When you want to pack up DNA really tightly so we can get ready to, split and move on in the cell cycle, that's where it comes into play. DNA, double helix DNA, then wraps around histones. Once DNA wraps around the histone, that generates a structure called the nucleosome. That matters in this. Histones are proteins that help in the packaging of DNA. Histones are positively charged, like we said before, and DNA is negatively charged. So the first bit is to go from DNA double helix by itself. Then DNA wrapping around histones. And remember for yourself that DNA wraps around histones twice, right? So the DNA wraps around each histone twice, so there's a lot of histones involved in this process. The nucleosomes then coil into a chromatin fiber, and then they further condense. And they start folding and they get folded, and that further condensation leads to chromatin. Now, there is, and you'll see this as we go on, H1, right? So it's H2A, H2B, H3, and H4. And when you start off like that, everybody's like, okay, well, where's H1, right? So H1 is called the linker histone. H1 binds to the external portion of the nucleosome and the linker DNA. So it's a linker histone. Why does this matter, right? Where will it come up? For us, it's going to come up in a few different spaces. One of those spaces is when it's time to understand which chromatin modifier can cause these different types of chromatin. We mentioned two types of chromatin, euchromatin, heterochromatin. Well, which chromatin modifiers can cause euchromatin or heterochromatin? And when we see DNA wrapped around our DNA condensing, how is DNA condensing? And that's really the question we're answering. DNA condensation matters so much in epigenetics. Okay, well, awesome. How is the DNA condensing? Well, the DNA is condensing by interacting with these histones. Well, the cool part about this is that those histones will play a role in how the DNA interacts with it. And that's what we're going to find out. As we start looking at the histones, there are certain histones. For instance, H3. H3 plays a major role in how DNA interacts with the histones on that nucleosome. So those histones are not just kind of, a ball that you need to know. I mean, a ball that, is just by itself, you need to know about it. No, those on those histones, right? On the histones that make up that nucleosome, H3 matters a lot. Actually, H3 and the lysines that are going to be attached to that H3 matters a ton. And we're going to show you exactly why that is. So as we go through this class, you'll start to see, okay, well, DNA interacts with histones, but the histone, the chromatin modifiers interact with the histones. And what they do to those histones matters in how DNA interacts with histones and whether you're going to have euchromatin or heterochromatin. And whether or not you have euchromatin or heterochromatin matters in gene expression. And that's what we're going to see as we move forward. How do we regulate gene expression from an epigenetic standpoint? This is one of those major base points that you have to understand. How the DNA interacts with the histones, and how does that play a role in how genes are expressed or not expressed? Hopefully this helps. If you have any questions, I am here throughout this whole entire course. You can meet with me at almost any point. So feel free, we'll have a group me. So feel free to hop in a group me, ask for an appointment with me if you want me to kind of break this down a little bit further. Feel free to reach out to me via email if you want to talk about this. I'm excited to see where we're headed this semester. All right, have a great day.